Welcome back to the course Fundamentals of General Immunology, and today we'll discuss some of the interesting facts about antibodies and its related biology, because the major proteins which we discuss most of our time in immunology is antibody. So we have looked into the different kinds of antibodies, how they function, what are the different ways they are classified, how they are linked to each other in terms of protein chains and all. So let's see some of the unique thing about antibody. So generally when people are studying about the protein function or the protein biology, generally proteins are degraded by use of any proteases. So proteases are the protein that helps to degrade the protein, and once the proteins are degraded by the proteases, the individual domain of the protein can be studied. And this is a very routine process which people in protein biochemistry does. So similarly here, if we concentrate on a very specific immunoglobulin, taking an example of an immunoglobulin, that's a gamma immunoglobulin, IgG. So we have looked into the structure of immunoglobulin G, where we have the heavy chain, this is the heavy one, and this is the light chain, and we have variable domain, and then we have a constant domain. So this is variable and this is constant domain. Then these two chains are joined by disulfide linkages here, and this makes the stable structure of the immunoglobulins. The size of the immunoglobulins are roughly 150 kilodalton. That's a unit of the protein weight. The heavy chain is mostly about 50 kilodalton. So each chain of heavy chain is approximately 15 kilodalton, 50. And the light chain is approximately 25 kilodalton. That makes to the total summation of 150 kilodalton. So as I told just a while, that proteins are degraded by proteases. There are many proteases which are available in the biological system, and one such protease is papain. So this is a very specific proteases that people use to digest the immunoglobulin and see what is happening. So when people added the papain at the pH about the neutral 6 to 8, and then what happened that after the treatment of the papain, the protease is very specifically cleaved to the protein site, and that created two fragments. So this fragment gets segregated. So that is what these two fragments are, which is actually 2 FAB, which is a fraction that can bind to antibody binding domain. So these are the FAB domain, and the bottom one, this gets segregated out, called crystallization fragment. So essentially these three fragments we can easily see when we treat the IgG with papain. Now so here these are the summary of the treatment. So when papain is added to the system, papain cleave above the hinge region in the disulfide bond, and then we can create 2 FAB and 1 FC fragment. Basically these are the FAB fragment, and one is the FC fragment, which is called crystallizable fragment. Again, this is a part of the heavy chain. Actually both are heavy, while this is a mixture of heavy and light chain. If you can see, one is coming from the heavy side, the other is from the light side. So essentially one can see that the FAB retain the antigen binding capacity. So basically even if we treat with the papain, we can have an antigen binding capacity of the protein, but the binding capacity is reduced to half since there are two fragmentation. So these can bind the antigen separately. So this is one small biology of the immunoglobulin G. When it is digested with a particular proteases, it can give us 3 fragment, 2 FAB and 1 crystallization FC fragment, which is approximately of the size 50 kilodalton. When you run a gel, we can see that since these are the size ranging from exactly the same, so we can see exactly the same dark or very thick intensity of the band at 50 kilodalton, that is mixture of 2 FAB and 1 FC fragment. So what is happening that if we run a gel, suppose this is a marker and this is uncleaved IgG, and if you said cleaved IgG, so this is a marker. For example, there are many bands, say this is 150, so this is a marker kilodalton that can give undigested fragment like here, and say if this is say 125, 100, 75, 50, then we can see that this after digestion we can see the band, and that intensity is very thick because this is a single band, and then here it is coming as a mixture of all this. So this could be the 2 FAB plus 1 FC fragment. So this is the combination of the degradation process. So what we understood is any protein can be studied in this way. Similarly, there is another proteases which is called pepsine. That's again coming from an enzyme that's secreted by human in stomach called pepsinogen. So this is an active form. Now if we see biology, stomach pH is acidic. That means these proteases can cleave the protein at an acidic pH, which is pH close to 2 to 4. So when we treat this antibody, same antibody with a pepsine at the pH of 2 to 4, we see a different pattern of degradation. What it will do? It will degrade into 2 FAB fragments, but these fragments are joined together with each other, and then 1 FC fragment. So what we are getting is actually 2 fragments, right? So one is having a combined fragment. Now just look into carefully, the earlier picture their fragmentation was separate, but here the fragmentation are intact. That means the binding capacity of this fragment is higher as compared to the earlier one. So the major difference between the papain and the pepsine digestion is the pepsine created FAB fragment is more active, and it can bind to 2 distinct antigen moiety because the complementarity determining regions are actually 2, so 1 plus 1, and therefore we can see that better binding affinity. Similarly, if we run a gel, then we can see that if we run exactly the same, so this will be a marker, and when we have undigested IgG and which digested IgG or cleaved IgG, and this will be treated with pepsine at acidic pH, what we'll get is, suppose this is 150 and this is say 100 and this is 50, what we'll get that in undigested fragment we are getting this 150 band, while in digested we can get 100 and 150. So we can see that clear cut in digested fragment, when it is digested with pepsine we can get clearly 2 fragments. This is actually 2 FAB, so this is actually called as FAB2, which is combined form, and then 1 we are getting same FC fragment. So basically the pattern of the protein is different when we are using a different proteases. So what it tells is when IgG is cleaved into 2 different environments, one is alkaline or neutral pH or towards the acidic pH, the pattern of fragmentation is different, and that decides the functionality of a protein. So the protein will be functional both way, but their binding ability will be different. So this is what people have tested different proteins in protein biochemistry, and this is one very particularly people are addressing or mentioning when we are discussing the IgG stability and IgG proteolytic activity. So remember, papain and pepsine are the 2 proteases which are used to cleave the IgG and understand how the fragmentation can work. So we'll move on and try to understand some of the unique thing about the heavy chain. So as we discussed that the immunoglobulin heavy chains are having both the variable region and the constant region, and particularly there is a region which is called hinge region, where we can see that this particular hinge region is what is giving fluidity or say flexibility to the arms of the immunoglobulin so that they can bind to different antigens. So regarding this hinge region, the immunoglobulin G, A and D, these possesses the hinge region, and therefore we see that the flexibility is more, so antigen can bind flexibly. And if we analyze the amino acid sequence in the hinge region, we see that the proline amino acid is really rich in that area, because proline can give kink to the native structure of the protein. While IgM and IgE, so we have 5 different immunoglobulins. Out of 3 they have hinge region, give flexibility, while IgM and IgG have an additional CH4 domain. So that means the hinge region is actually masked by an additional constant region in the Hebby chain, and therefore these particular immunoglobulins IgM and IgE lacks hinge region, and therefore we don't see the structure to be flexible, and therefore we can call them, they are more rigid as compared to the IgA, G and D. So remember, IgG, A and D are little flexible because of the presence of hinge region, while IgM and E are rigid because of the absence of hinge region. And why there is an absence of hinge? Because there is a additional CH domain in the hinge region. So the hinge region is occupied additionally by a constant region. Now when we see the FC region, this FC region is also very important, and this FC region can help us to understand few important things about the immunoglobulins. So first thing is how this FC, so which FC receptor the antibody will bind will largely depend upon the amino acid sequence present in this constant region. Secondly, the complement activation depend upon the amino acid sequence present in the FC region. Also, the half-life of a protein, say for example half-life of IgG, half-life of IgA and D, other immunoglobulins, will be decided based on the FC region's amino acid sequences. So this is very important to decide the half-life of a protein. How it is distributed, that's also very important. So these immunoglobulins are distributed in blood or mucus. For example, IgA is more in the mucus area or more in the secretory area. How these are different as compared to blood specific immunoglobulins or the placenta specific immunoglobulin depend upon this FC region amino acid sequences. So defining this is very important. Secondly, since we know that these immunoglobulins are actually bound with the disulfide linkage, basically they are either monomer, dimer or pentamer. So these are also decided based on the sequence of these FC region, because the J chain that's help in the binding of those monomeric form to pentameric form as in case of IgM, and to the dimeric form as in case of IgA, is actually depend upon the J chain which can bind to the FC region. So the sequence of FC region, the conformation of FC region is very important to decide all these parameters. So before going to the details of the antibodies and all, let's try to discuss very important discoveries that happen in antibody field. So when we see the 1984 Nobel Prize in Physiology and Medicine, the prizes have been given to 3 scientists in combination. So one is Dr. Neil Jorn, who invented different specificity in the development and control of the immune system in the immunoglobulins and also overall immune system. While the other 2 scientists, Kohler and Milstein, they got a Nobel Prize for the discovery of monoclonal antibody discovery. When we say monoclonal, mono stands for single, clonal means a clone of a cell. That means if a particular cell is selected to secrete a single kind of immunoglobulin, we call it monoclonal, that's coming from a single clone. If the antibodies are coming from 2 or 3 different cellular population, then we call it polyclonal. That means we have either polyclonal, that's coming from multiple cell type, and if it is coming from a single cell, we call it monoclonal. That means antibody, that's a very specifically a similar kind of structure and conformation. The biology of monoclonal antibody was invented and discovered by these two scientists, and therefore they have been awarded for this prize in Physiology and Medicine in 1984. It's a great discovery. We understood the use of monoclonal antibody in therapeutics and its application nowadays very rampantly in many areas. So let's try to see what do you mean by monoclonal antibody, how they are produced. So the monoclonal antibody is produced by a technique called hybridoma. Hybridoma stands for the name. If we mix two terms, one is called hybrid, that means a mixture, hybrid means two. Oma stands for anything that's coming from cancer. Melanoma, that's cancer of skin. The hepatoma is a cancer of liver. So oma, whenever there is a suffix oma, that stands for cancer. That means the technique which is utilized to produce monoclonal antibody is a mixture, and in that mixture one of the cell is coming from cancerous mass. That's the meaning of hybridoma. So what is the process? That we generally, if you want to develop a monoclonal antibody against an antigen, so we need to select an antigen. For example, a protein, say any protein, for example the SARS coronavirus, a spike protein. So if we take a spike protein of SARS coronavirus and if we inoculate into a mice, essentially we call those antigen as an immunogen. So that mice, when we inoculate S, the antigen will go inside and can start developing an immune response. And mostly people used to collect the spleen. Spleen is called graveyard of RBC, and ultimately the area where we can get lot of B cell. So the immunized mice will be sacrificed and the spleen of those animals are collected, and from the spleen we could isolate the B cell from the cellular mass, and once we isolate the B cell, then we can take separately some of the cancerous cells. Now we know that the cancerous cells are immortal, so they have a capacity to multiply, and the B cell is known to produce an antibody against the spike protein of SARS coronavirus 2, but they have a finite lifespan. They cannot replicate throughout the life, and therefore we know that there is a B cell which can create an antibody that can bind to the spike protein of coronavirus, but then they can't survive for a longer duration of time. So if you want to make a cell that can continuously produce the antibody of our choice, then we need to mix this cancerous mass along with this. So once we take the cancerous cell and fuse it with the B cell producing spleen cell, we call it splenocyte mixture, or mixed with the myeloma cell, then we could see that there could be possibility that the two cells can fuse together with each other. And once they are fused in the presence of certain chemicals such as polyethylene glycol, PEG, so polyethylene glycol will help to fuse these two cell mass, and once there is a fusion, the product is called hybridoma. So that means once we get these two cells fused, we call those cells as hybridoma. And once the hybridoma cells are produced, that means these cells are having both the properties. They can create a antibody, they can produce an antibody because they are already immunized with the spike protein, and they have a capacity to multiply throughout life, so they won't die. But then we need to select, because essentially when we have mixture of these B cell and the hybridoma, myeloma cell and the hybridoma cell, in a mixture we have all the three cells. We need to select only the hybridoma cell, not the B cell, not the cancerous cell, because essentially we need to make a therapeutic intervention, or we need to select the cells which are actually true hybridoma. For that we need to use a media called HAT media. The biology of the HAT media I will just explain in the next slide, but for time being just try to get an idea that HAT is a selective media. It's a very, very selective media which stands for hypoxanthine, aminopterin and thymidine. So this is a mixture where only hybridoma cell can survive, and both the myeloma cell and the B cell which are coming from a splenocyte, we can call it splenocyte, they all will die. So the splenocyte will die, because of the hat media. The myeloma cell will die because of the hat media. The biology we'll see in a while, and the only cell which will survive in this hat media is hybridoma cell. So this we call a selective media. The selective media for hybridoma screening is hat media. And once we select the hybridoma, then we can plate it into a 96-well plate format, and we'll try to clone those, or try to say increase the cellular mass and see that what are the things which are coming out from the cells. So suppose if we seed all the 96-well plate with all the cells, and we can collect the antibody after a certain interval time, we can identify that there are multiple hybridoma cells, and each cell is producing different quantity of the immunoglobulins, which are very selectively binding to the spike protein. So we can titrate the immunoglobulin with its binding affinity. So we know that this particular well is producing an antibody that can bind to the spike protein more effectively as compared to the other. We can take out this hybridoma cell, grow it in a bulk, and then store it into liquid nitrogen, Ln2, which is minus 196 degrees centigrade, and then can store for the future use. What will happen, that we have generated a hybridoma cell that's having a character that they can produce a antibody that can bind to the spike protein of coronavirus, and essentially whenever we want it, we can generate a huge amount of antibody from those hybridoma cells. So this kind of a factory which can give us a ready-made produced antibody whenever we want. This can be utilized for therapeutic intervention, they can be utilized for the research purpose. So this is the groundbreaking research which Kohler and Melstein did, and tried to give us a very specific, the cell was which can create a very unique monoclonal antibody. Since these are coming from a single B cell population, a single splenocyte fused with the melanoma cell, then therefore we called it a monoclonal. It's just coming from a very single type. We can characterize, and we can use it for further, say, biological intervention. So till now we understood how these monoclonal antibodies are produced. Let's try to see what is HAT and how this whole biology takes place in this hybridoma technique. So there are two pathways of nucleotide synthesis. One is called de novo and the other is called salvage. So there are two pathways which always takes place in the human body and also in other animal, where the nucleotide are synthesized from the scratch. Say we can take out the precursors such as amino acid, glucose, carbon dioxide, etc., to be made for very specifically nucleotide sequences which can be utilized for the DNA synthesis. That means once we start from the scratch individual component that can make an amino acid, such as the amino acid pool, the glucose and the CO2, then we can call that pathway as de novo pathway. If there is a degradation, if the bigger protein are degraded, then we can always see that there is a recycling of the compounds. If there is a recycling, we can still do a synthesis of the same molecule, but there is no scratch biology. It can start from a ready-made bigger moieties, and therefore these are called salvage pathway, which are started, which are creating the bigger picture from the scratch molecule, which are degraded out. So salvage pathway is basically coming from the one which is already degraded. So if we see, there is an hypoxanthine, guanine and thymidine. So these are the three nucleotides which are, with the help of two enzymes called HGPRT, that is called hypoxanthine guanine phosphoribosyl transferase, that can convert into individual nucleotides and that can be utilized for the DNA synthesis. Similarly, thymidine can also convert it into thymidine monophosphate or diphosphate and further triphosphate by thymidine kinase, because kinase is actually what will eventually do phosphorylation, and once there is a mono, di and triphosphate, basically the one which can be utilized for the DNA synthesis. That means these two are the pathways which are utilized for nucleotide biosynthesis. Now what is HAT is the component, as I discussed. The H stands for hypoxanthine, A stands for aminopterin and T stands for thymidine. Now hypoxanthine is actually a purine precursor for the salvage pathway, which we have just now studied, and aminopterin is actually inhibitor of DHFR. So DHFR is dihydrofolate reductase, which is very critical enzyme that is required to convert the precursor of the amino acid and to be converted into the bigger chain. So they can inhibit the DHFR and block the de novo synthesis pathway. Thymidine is actually the source of thymidine, which is actually utilized by thymidine kinase to convert into DNA synthesis. That means we are providing two components that is helping in the salvage pathway and the one component that can inhibit the de novo pathway. What is the beauty about the HAT media in selecting hybridoma is given below. So what's happening is the normal B lymphocyte, which can generate the antibody, is positive. They contains a GPRT and thymidine kinase. That means these two enzymes are present in this cell and therefore they can do salvage pathway. So they are utilizing the salvage pathway and they can generate this antibody. However, these cells are immortal. No, they are mortal. They can die, right? So they won't survive for a longer duration of time. Therefore, after a certain period of time, the cell will die. But the myeloma cell, which is actually a GPRT negative, thymidine kinase positive, and they are of course immortal, so they can multiply for a longer duration of time. Since we are using HAT media to screen them, the de novo pathway, because the only pathway which left out for these particular cell is actually de novo pathway, because the GPRT is negative, so they can't go with the salvage pathway. So the only pathway which myeloma cell will require is a de novo pathway. Since we are using aminopterin, which can inhibit the de novo pathway, we won't see anything happen in the myeloma cell when they are growing it in the HAT media. That means in the HAT media, myeloma cell will die. The lymphocytes will survive for a while, but again, since they are not immortal, they can die, because after some time they can multiply and after some time they will eventually die. This cell will also not survive for a longer duration time. While the hybridoma cell, which have both the property, they have the GPRT positive, thymidine kinase positive and immortal also. So they can do both salvage pathway as well and the de novo pathway as well. And also, since they are immortal, they won't die, and therefore the beauty of this biology has been explored to produce a cell type which is permanently produce the immunoglobulin. That is what is needed for the hybridoma technology. So this is all about when we are trying to understand the biology of the HAT media that's utilized for producing a monoclonal antibody. Now let's try to understand some new concept on monoclonal antibodies. And since we know that the monoclonal antibodies are of great use, they are utilized for producing something very unique. They can bind to a specific antigen and they can produce some function. There are two terms which we are going to define here. One is called chimeric antibodies and the other is called humanized antibody. When we call chimeric, it's a kind of a mixture. Chimera means mixture of two. Humanized means the one which looks like human. So let's try to focus on the two antibodies here. So this is actually the orange antibody that comes from a mice variety, and this is the form which is coming from human. So if we take human and the mice, these antibodies are different. Now this human antibody is converted into humanized, and this mice antibody can be converted into chimeric antibody. How it can be achieved, we will try to understand that. So these are the two variations in the monoclonal antibody. So these can be utilized for many other functions. And what's happening that when we make a chimera, that means this portion is coming from human. So if we take this portion from the human and only the complete upper area, which are the variable region of the constant and heavy chain, is coming from a mice, then we call it a chimeric antibody. While we can understand that this is a mixture of two antibodies, one is human, the other is mice. Now this mice specific area in the heavy and light chain that's binding to the antigen can give specificity, and therefore people are using it for some therapeutic intervention or some kind of an use that can be done or utilized for the human medicine. We'll see in a while some of the drugs which are available in the market utilize the chimeric antibody as well. While in case of humanized antibody, everything is of human except the complementarity determining areas which can bind to very specifically epitope of an antigen is coming from mice. That means we can see that the humanized antibodies, actually everything is human except the part of the complementarity determining region which is coming from the mice species, while in chimeric we see a bigger fraction of the mice antibody. That means if we try to compare the chimeric and humanized, more content of the human immunoglobulins are coming into humanized, while the lesser content is coming into chimeric. While if we consider the mouse antibody part, more amount is coming into chimeric while less amount is coming into humanized. Remember one thing, that complete heavy variable site of heavy and light chain is coming into chimeric, while only the complementarity determining region CDR that can bind very specifically to the epitope is what is available in the humanized. In terms of nomenclature, the chimeric antibodies are always end with CMAP, and the humanized antibody is always end with SHUMAPs. So these are the two very unique way people are doing a nomenclature, so one can always understand that whether this is a humanized antibody or chimeric antibody. So if the drug name end with as a XIMAB, we can understand this is a chimeric, while if it is coming and ends with ZUMAB, then we can understand that this is coming from a humanized antibody pool. So this is what the drug which are available in the market. One is called rituximab. So rituximab is target for CD20 protein. So basically we can create a mice antibody that can bind to very specifically CD20 protein. Since we have to use it for the human trials, essentially we can make a chimeric antibody so that the larger fraction is coming from the human, and therefore we see a less immune response against those proteins, because again these proteins are from outside. We always see that these can behave as an antigen. To avoid those antigenicity, we can create a chimeric antibody. So this can be target for CD20 protein, and that to treat non-Hodgkin's lymphoma. This is of different forms of the cancer which is actually called lymphoma. So lymphomas are two types. One is called Hodgkin's lymphoma, the other is called non-Hodgkin's lymphoma, and also in chronic lymphocytic leukemia. So this is a blood cancer. So essentially in these two forms we see that the CD20 protein expression is enhanced. So if you can use some of the antibody that can bind to CD20, we can see that the effect is getting nullified. The other is actually inflimizumab. Inflimizumab actually is a TNF alpha neutralizing antibody. So they can bind to TNF alpha, which is a tumor necrosis factor, and this is utilized to treat autoimmune condition like rheumatoid arthritis, Crohn's disease, and ulcerative colitis. So that means this TNF alpha is getting higher concentration in the patient who are suffering from either Crohn's disease or ulcerative colitis or rheumatoid arthritis. So just to subside or downregulate the TNF alpha concentration, we can see that we can use some of the chimeric antibody and can see that if we can mask TNF alpha or CD20 for that matter. So these are all about chimeric drugs available in the market. There are many other drugs. I am just giving few examples here just to give you an understanding, and always remember, so these are XIMAB, stands for these are all chimeric drugs. While trastuzumab, which is Herceptin, is a drug available in the market that's to treat breast cancer. So this is actually a humanized form of the antibody. We can see that this ends with ZUMAB and it inhibits the HER2 receptor. So this is human epidermal growth factor receptor 2, and that's a very specific for some of the breast cancer which are really aggressive. They can do metastasis and go to different parts of the body and spread the cancerous mass. We can use humanized antibody to treat those aggressive form of the cancer. Then we have something called pambroli. So this is pembrolizumab that can block the PD-1 pathway. We have looked into PD-1, which is a programmed death domain 1. So if these proteins are expressed in a particular cell type, which can bind to PD-1. So basically PD-1 can recognize by PD-1 ligand, and essentially there is no immune response against cancer cell. So if we can block the PD-1, then essentially the immune response can fight against the cancer and clear the cancerous mass from the body, and therefore the body immune system can recognize and fast fight with the cancer cell if we can mask the PD-1 specific domain. And therefore these are some of the antibodies which are available in the market that can be utilized. So in general we can understand that monoclonal antibodies which are created and identified and discovered by Kohler and Melstein has a wide application. We can create them by using some of the very unique technique called hybridoma. We can select it by hat media, and after making this monoclonal antibody there are many variations which are available. One is called chimeric, the other is called humanized. Chimeric can be utilized for many disease condition when this can be mixed with a human antibody with a receptor coming from the mice antibody, and also we can do a humanized antibody where the complete human antibody is doped with CDR from the mice, and therefore we can utilize it for further say application in the drug area. So this is all about some of the interesting facts about the antibodies and some of the unique combination which are available in the market that can be utilized for therapeutic intervention. Thank you.